Yeah. How are you guys? How is everyone? Good. Well, unfortunately, Ms. Lindemann has come down with COVID and hopefully not transferred it to me because we were hanging out together for a bit yesterday. But we are gonna continue on. So today in lab, in person, there were two reasons. Well, there was only one reason it was in person, and that was because you had to treat your next set of cells, right? So how many times have we treated ourselves already? By the way, remind me. Okay, yes, we did only treat them once. How many sets did we treat? So how many times have we done like kind of an entire experiment? How many sets did we treat? Three. How did we do three? Oh, I know what you're thinking. Um, yeah, so in this slide, in your, if you're going by that, right? So we did it in the 6-well plate where you had the duplicates, right? And that set was used for what type of analysis? Cell viability. So we have done one set where the cells were treated in duplicate and we analyzed it for viability analysis with the trypan blue and the starred confluence. And then we ran another set, which was how were those plated? How were the cells plated in that one? That's the stuff that you examined last week. So how were the cells plated in that set? Were they plated on the petri dishes? So there were cover slips inside. And again, we did that in duplicate, right? So there were two individual cover slips. The petri dish was just a holder for them, and each cover slip was kind of our replicate. So we did one set in duplicate where it was analyzed for viability, and then we did the second set in duplicate where the cells were on cover slips, and we used that for what type of analysis? For the morphology experiment. Exactly. So we stained the cover slips and then we made some morphological analyses, right? Some morphology observations on them, some measurements on them. And what program did we use to do it? Yes. So we use the ImageJ. And by using ImageJ, we are able to kind of take out our bias a little bit, right? Among other things. And we can do more sophisticated analyses than our naked eye can do, right? Because we can do things like area and we can do things like perimeter, right? So there are some things that we can do that we could not have done, or like the circularity, right? The shape descriptors and stuff that we could not have done using just our eyes. So far, so good. So today we would have plated, you would have gotten another set of plated cells and you would have treated them a third time. That was the first part of the lab. So we are going to fake treat them today, you know, in silico, and then I will go and treat them for you in real life. So let's go through, first of all, that process so everyone is on the same page about what it is that we do when we treat ourselves and we remind ourselves how we do it. And then we are going to do our calculations that we would need to do for today if we were treating ourselves. We are going to turn in that assignment. So I have that information. So I can do that for you when I go into the lab today. And then we will start on the Excel portion. Okay? So we will, let's see if I say you. Is it sharing the whiteboard? Perfect. Okay, so let's do it. What's going to be our step one? What do we do when, first of all, let's remind ourselves, if we are uh, treating cells, right, in culture, do we need aseptic space? Yes or no? Yep. So we are going to need aseptic space. So we're going to need a flame, and we're going to clean our area with conflict and all that good stuff. Yeah. So once we set up aseptic space, what would we want to do next? You guys can talk too, by the way. You don't have to write in the chat only, you know. So you want to collect your supplies and have a beaker with conflict. What is the beaker with conflict for? Yes, it's to collect our waste. And what's the purpose of conflict?
Sterilize.
Huh? What does it do? Say it again. Sterilize your, or decontaminate, not sterilize necessarily, but decontaminate your liquid waste. That's our purpose with that, right? I want to move you a little bit. So collect supplies and set up um, aseptic space. Okay, next, what do we do next? Oh no, why is that line over there? What is the purpose of this line? Where? Sure, you can tape it to remind yourself what that space is. That would be part of setting up your aseptic space. You don't have to tape it, right? We could just, once we are used to it, we kind of have a mental picture of what area is aseptic, but you could definitely tape that space to remind yourself where you should be working and how you should not go outside that area to maintain aseptic treatments. Now, what are we gonna do? Gathering materials, wiping up your supplies, sanitize hands, equipment, square with tape. All this are part of setting up aseptic space and collect supplies. And yes, those are all the things that you would do. What are you checking concurrency for? You never got the cells. And do you really want to get your cells yet? Because remember, you're on a timer once the cells come out. If you remember last time, we had a lot of issues with infection, and one of the reason was that they were staying out too long. Typically want to keep them out as little as possible, right? And so technically you can do this procedure, actual procedure with the cells in like 5 minutes, right? If that. So you don't want to take them out yet. Caitlin, so you said you want to prepare treatments. Yes, we've already switched on Bunsen burner because we set up the aseptic space. Yes, you are going to collect your treatments and you're going to prepare your treatments. So for today, you have 3 plates only. So you're not doing it in duplicates. You're just doing a single set of small plates. You have an untreated plate, a plate that you're going to treat with your whatever chosen natural supplement you have, and a third plate that you're going to use your active compound on. Cool. So you are going to prepare your treatments. That's correct. So to prepare treatments, how many, what should you get to prepare your treatments? Go ahead and write. Prepare. And where do you prepare these treatments? Prepare treatments where? In the plate, in a beaker, in a tube. So you need your chosen active compound and natural supplement, and where are you going to use it? Are you just going to use it straight up? What type of tubes are you going to use? Mini tubes, like 0.6 ml? Which size centrifuge tubes? Yeah, they're 15 ml ones. So in 15 ml conical tubes, right? But we only need to prepare for one plate. So it's 3 ml of media, 3 ml of fresh media in 15 ml conical tubes. So you would have gotten today 9 ml in one tube. So what you would do is you would take 2 extra tubes, right? So I will actually draw that out so you see it. So you would have gotten one tube with 9 ml of media. And then you would have gotten 2 new tubes and taken 3 ml in each one. So you would have been left with 3 ml in the last tube. And so then this one would have been your untreated, and then you would have your natural supplement and your active component. Okay? So, okay, you went ahead, you prepared your treatment, right? Untreated, natural supplement, active component. We got them. Now, what do we do? What's next? Now you get the cells and check your star confluence. So you are going to, first of all, I will draw, write it out, then I will draw it. So you're gonna select cells and record start confluence. Okay. Which type of microscope are we going to use for this part? The inverted microscope. Good. So it's an evil eye. That just meant I'm going to be checking them under the inverted microscope. And what you're checking for is health, right? You're checking for infection, and you're checking for start confluence. Exactly. So you're making sure there are not too many floaters. You're making sure the cell is okay. They don't look like there are infection particles in there, like bacteria or yeast or fungus growing inside. And then you record your start confluence. And you make sure that the cells look like the type of cells that you've had in the past. Because there are other cell lines in there and you can get a different one by accident. Okay, now what will we do next? Yeah, so the first thing we're gonna do is we're gonna remove the media, spent media. and disposing conflict beaker to disinfect. Got it. What after that? What do we do next? We are going to wash with PBS. Do we remove the wash? Sterile PBS, and then remove all PBS and discard in waste beaker. Okay, very important that we need to remove the PBS as well. We cannot let it hang out in there. What happened to my arrow? What was the sound arrow? Okay, then what? We're gonna label our plates along the side, not on the lids, but on the side, so it's not going to be switched by accident. So we are going to label plates for each treatment on the side or bottom along with our group info. And then we are going to add appropriate media, treatment media to each plate. And then what do we do after we've done all this? What do we do now? We added the treatment, so that's what I wrote about this. Add appropriate media to each plate. So we added the treatment media. Now what? We've added it. They are now getting treated with the media. Now what do we do? Yeah, return plates to the incubator and clean up aseptic space, reset bins. One more thing that I did not uh, remind you of. So when we prepare treatments, how do we add the drugs to them? How do we add the drugs to our medias, the treatment tubes? What do we use? Well, you're putting them in the tube, so it doesn't really matter whether you're shooting it directly on or not, but there is something. Which type of pipette? Sterile micropipette, exactly. And where do you find sterile micropipette? Where do you find the sterile tips? Nice. Exactly. So add treatments to tubes using sterile tips, autoclave tips. The line tape is basically that they have been autoclaved to sterilize. Autoclave tips from the instructor guides. Cool. Okay. That went a lot quicker than if you were there. No? Funny. Very good. So good? We got everything? Got the gist of what it is that we were going to be doing? Now we need to do the next part. No, it's not based on confluency. It's based on the concentration of the drug that you need in the total volume. And yes, that's something you would have done already when you set up the aseptic space. So the first thing you would have done would have been that you calculated the compound, how much compound you're going to be adding, fill out that sheet, and then you would have made this little chart like I've made before you got your cells. So we're going to actually do that next. We are going to make sure that we have the amount of supplement that you need or amount of drug you need to add to each treatment correctly calculated so we can um, make sure that I am treating them the way you would have. Okay? So remember, I'm just your technician today. So if you measure wrong or calculate wrong, then we would be in trouble. Yeah? Okay, I'm gonna stop this share first. How do I always forget how to close out the whiteboard? I guess it's here. Okay, there we go. So let's go ahead and do that calculation now. So I'm gonna share my screen now so I can show you where you're going to find that information. Share. And we are going to go to this assignment that you have. So go under your assignments and can you see my screen, guys? Yes? Okay, perfect. So we are going to go to week 9, treatment 2, experimental design. Now, it tells you that you have only one 160 millimeter square plate per condition. So you'll have 3 plates total. The first one is untreated, so obviously you don't need any volume of drug in there. You just needed to have taken the start confluence. And then the other 2 will be your natural supplement or active compound. I do not want you to write natural supplement and active compound. I need you to write the name of the natural supplement and the name of the active compound. Where do you find the rest of this information? This is the volume of drug that you will add in 3 milliliters of media. Okay? So you're going to go back to your, where is it? Lab and modules. And once you're there, you are going to this week's module right here. And right here is the treatment day protocol, viability morphology, right? So we already know we are modifying it for the 3 ml volume. So we just have one. That's why we made that little chart that we just did. You're going to open this up. And in here, you will find these concentrations. So this is where you get the stock concentration of your drug. And you also get how much of this drug you're adding in. Guess what? I already tell you how much you need to add in 3 ml of media. Now, what I would like you to do is to make sure that these numbers are correct. And the way you do that is by calculating the final concentration that you will get of this drug in your 3 ml media when you add, for example, 22.2 microliters of ashwagandha in 3 ml. When you calculate it, it should match up with the last time that you did it. So when you did treatment 1 and 2 and did the experimental design for that, it should match up exactly with the final concentration of that. Okay? So what I'm gonna do is give you guys, let me see, where did the stop share go? Stop share for a second. I'm gonna open up your breakout room. Each group, you just go to the station that you belong to. So if you're station 1, go to breakout room number 1. Station 2, go to breakout room number 2, and share your screen in there with each other and do this assignment together. Okay? You'll have 15 minutes to work on it total. If you have a question, it should not take you that long, but I'm just giving you that total amount. When you're done, you submit it and you come back to the main page. If you have a question, raise your hand and I'll come and help you. And then once we're done with this, we will go ahead and start on our statistics. Okay? So breakout room, they are just 1 through 12, so you're just gonna go to the one that you need to. If you need help getting to that room, if you don't see the, join button, let me know and I'll place you where you need to go. So there you go, they're open and you can join. I'm pausing the recording for now. Please remind me to restart it when we come back to the main page. So given 3 pieces of information, you should be able to figure out the last missing value, right? So what we are gonna be doing here, C1 is gonna be the stock concentration of your drug, which is listed in one of the columns, okay? 1 second. Then V1 is gonna be V1 is going to be the volume of drug you need based on that chart for 3 ml. Okay. C2 is going to be your X in this case, right? Which is final working concentration of drug that the cells receive. And V2 is going to equals 3 ml. Most of our drugs are reconstituted in alcohol, which evaporates out. So your final concentration or the final volume pretty much remains that 3 ml. Okay? You good, Blue? So that's basically the gist of how you're calculating that concentration. Go ahead and calculate it since you didn't do it the first time. Who's treated yourselves the first time? I don't see the chat anymore. I don't know where it went. And I can see it. So if you're saying something, can you please actually say it so I can hear it? Or write it on the whiteboard. Kathleen, what you doing? I see your little pointer moving around. It's fun. You're playing mouse tag. Okay, I'm gonna go.
For active compound, the stock is 10 millimolar and the suggested volume in microliters is 2 microliters. Do I convert the 2 microliters to 0.020 milliliters?
Do the other way around. Convert the, yeah, you can, but it's probably easier to convert the milliliter into microliter, which is 3,000.
Okay.
You know what I mean? Yeah,
Yes,
you
ma'am.
can go either way. You can convert it into milliliters if that's easier for you. It would be what, 0.002, right?
Yes, I'll just
As it-
convert the milliliters to microliters.
Yeah, one or the other, as long as you have them in the same units, you will be fine. But for molarity, you have to convert to make sure everything is in the same units. Okay?
Okay,
I'm gonna
thank you.
close all the rooms so people are back in the main room doing
Thank you.
something. And then we can start on the next part. For those of you who are ready to move on, well, actually, let me have all these people join back in 43 seconds, and then I will tell you what you can work on, start working on in the meantime.
I have a I have a question. I was wondering for the suggested volume, do you use either the suggested volume in 1 milliliter or the second column that says in 3 milliliters?
So remember, guys, if you are using the column in 1 ml, you have to multiply by 3 before you get the actual amount of drug that you're going to be using in your 3 ml of total volume. So why not act smart and just use the one that's in 3 ml? It should be exactly the same once you do the conversion, right? Did that make sense, Evan? Or Vito, whoever asked the question, who asked the question?
Yes, thank you.
Okay, so everyone good? Everyone able to get this done? Or almost done at this point, hopefully. Just make sure that you submit it before you leave the Zoom today, because I will be working on that today in the afternoon, okay? Okay, so let me go ahead and close this out. And let me tell you what we're gonna work on next. So next, what you're going to do is we're gonna be spending the rest of time learning about statistics and how we can get meaningful data from or meaningful conclusions from the data that we've collected. I know some of you had infection and weren't able to use your own slides, but at the end of the day, you did choose to use certain images from the backup data, and you did make all your own observations from them. So it is your data, and you should be owning it. And so what we're going to do now is we are going to look at that data that you've collected, and that is going to be contained. So you're going to need to download a few of your older assignments so you can collect that data from there. So what the first assignment that you're going to download or that you will open up on your screens is the viability assignment. So this one where you turned in the viability data, right? So this is what it would look like. Let me just remind you, this one, yeah, that's the first one that you're going to need because the raw numbers are now what we're going to be looking at. And then the second one that you're going to be opening up is going to be your morphology assignment that you just finished collecting data for and that you turned in recently. So that's going to be the one over here with all this information right here. Okay. So that's what you're going to do. And then where we are going to go with this stuff is going to be the assignment for this week, which is our somewhere in here, down here. Data analysis and statistics, this one. And so you're going to download this data analysis and statistics template. Let me make sure this is the correct one because I had given them an updated version. Yep, looks like it. So we are done. So go ahead and download that. Now I already have them downloaded. And so on my end, so I can show you what to do. What I want you to, what you're going to be doing is you're going to take all your raw data and you are going to be placing it inside this template, okay? And that means that you want to make sure you're putting them in the correct place. Now, if you had numbers that were handwritten, then you'll obviously need to type those numbers in here. If you had numbers that were not handwritten, that were in the actual document typed up, you should be able to copy paste them. Sometimes when you copy paste, your numbers had some formatting issue which causes it not to work properly. So you may need to, at that time, fix that and I can help you. But for now, either copy or hand type, if they were handwritten notes, type in the information. The ones that we're going to work on the first as a class is going to be our relative growth. So I want you to, and tripe and blue. So I want everyone to make sure that those are the first ones. Those are the ones you prioritize. But I will show you how you can go through all of this in just a second. Let me open my viability document so I can do that. My viability document is handwritten as well. So if you have things that are handwritten, like I said, then you would be copy pasting it. Okay, so let's go to the relative growth. The first one is gonna be this information, right? So the viability two sets, those are referring to these two sets. So you had two untreated, two of your natural compound, and then two of your active compound. So what you would be doing here would be writing on top what they were, saffron, frozen, for example, right? And then in here, I'm gonna write in my start confluence. So I'm just copying. I cannot copy apparently. Saffron 25 and 30, and then frozen 35 and 35. And then over here, I'm gonna do my end confluence. Now, one of the things I can do to make sure my labels get copied correctly is do something like this. This is just a trick that you can do to make sure you don't have to type as much, right? I can do the same here. Okay. Now, let's go ahead and you're gonna put in these numbers for the end confluence for this one. And so I have 90 and 70, or this is 40. I was correct, never mind. So then it was 90, 75, 25, 20. This is in the data analysis and statistics assignment from today. So that's something that is in this week's module. I'll show you again in just a second. 25, 20, and 22. Okay. So where you find this spreadsheet is in this assignment, data analysis and statistics. It's a 70-point assignment. It looks like this. And this is the sheet that you're taking out. Okay. Okay, so now where do I get the other two pieces of information? Well, I get the second set of information from the morphology assignment. Now, some of you may only have one set of data for morphology, like you only looked at one of the cover slips and copied that morphology. That's okay, whatever, that's fine too. You don't need to have both numbers as long as you have at least one set. You are going to be showing me, you're going to have the minimum of n equals 3 for statistics. So don't panic and just use that. I'm trying to find my morphology. Where did my morphology go? Where did I put it? Oh, it's right here. Never mind. So yeah, so for morphology, again, I'm gonna do the same thing. I'm gonna write down what comes, I had for one or the two of my numbers. Let's say I only have one. In my case, then I'm gonna say, 6. I will put that in, okay? So that's how it's gonna look like. Now, if you look in here, this has a formula in there already, and this is meant to be able for you to convert it into relative growth. We're going to make sure that formula is done correctly, and so on purpose I have left it a little bit loose here. I am recording, right? I want to make sure. Yes, yes. Okay, so you're going to write equal sign and one equal sign, right? I'm just confirming. And you're going to have your end confluence divided by your start confluence. And because for each cell, it's going to be its own thing, right? Each time it's the end confluence of that particular type divided by the start confluence of that plate. That's going to be the way you do it. And then you can just make sure all those numbers are correct by dragging them over and down. So that's going to be your first set. You're then going to go to your trypan blue, and this is going to be the trypan blue numbers that you've got, right? So I have mine in another sheet from last semester. That's what yours are going to look like once you're done with everything. But for now, you're not worrying about that. I'm just going to collect my numbers from there. And if you're collecting numbers from data that's already typed, you should be able to just copy paste it. You're only going to copy and paste the number of blue and the number of clear. Do not copy over your totals. So I'm going to take my untreated, Control V. My saffron. And then my closing. These are gonna be my saffron and frozen. And here it's gonna be same thing, saffron, frozen. And I have my numbers. And then what you want to do is take these numbers and you want to reduce, oh no, the number of significant figures to no more than 3. And that just simplifies the data so it's easier on your eyes and you can see trends a little bit better. I like to make them centered. It makes my life easier, but you don't have to. Yes?
I have a question. I was wondering how can you show again how you reduce the sig figs?
Yes. So you select it where you want, and on your toolbar, there should be something like this where you can like decrease or increase depending on which direction you go. If you don't see that, you can also go to format cells and you can add the decimal places over here as well to whatever you want. Okay?
Okay.
Make it into a number and do it that way. Yeah, but I have the
Okay. Thank you.
You're welcome. Okay, so we'll talk about analysis later, but now the next part is going to be your counts. And now, I have it already designed so that it should technically go ahead and put all your counts, the names correctly. This one is the only one that didn't. I don't know why. That's funny. E7, so this one is supposed to be E. So yeah, so there was a mistake over there. And again, now in this case, you're going to be looking at your morphology assignment, right? So for example, I'll just show you what I have from another semester. Oh, that's not a, where did it go? This one, right? And so that's what you're going to be copying, your numbers that are in here, okay? And again, you're gonna have 4 sets of numbers. Oh, wrong thing. You stay here. Uh, mhm, and we can do the string. There you go. And you're gonna do the same thing here, right? So you're gonna put in your data. Um, and this is where sometimes it can have its mind of its own. I haven't figured out what make it one way or another, but sometimes it doesn't like it and it does weird things. So just be aware. Saffron is right here. So saffron. And I'm literally copy pasting for now. So, and then I have this actually isn't frozen. This is Taxol, so it's showing the effect here, but yeah, that's okay. And then you're gonna do the number of nuclei the same way. You just do not do anything to the bottom. Keep it the way it is. Do not change this formula. It already has a formula. We'll talk about the significance of this formula later. Only put in your raw numbers, okay? And then you put in the dividing and all that. For your cell measurements, I would like you to not put the information that you have in micrometers, but only the raw pixel values that you got from your results, okay? So you only need to put those in for cell body diameters and nuclear diameters. And then finally, in your interesting observation, if you look at this one, you notice that you have different options available. Now, if you have something interesting that involved measurements, maybe you looked at length of projections, you looked at length of, you like changes in the shape of cells, you looked at perimeter, area, anything like that that involves measurement, then you'd have like 20 numbers in there, right? Because you did 5 from each field. So you may have 15 to 20 numbers there. Those numbers for all the untreated, all of them, all the fields, just put them one on top of the other. Then you'll have your saffron numbers, Fossa number, right? So your treatments. If you don't have measurements and instead you did either percent multinucleated or you counted number of vacuole or number of cells with vacuoles, number of cells with that are differentiated, number of cells that are apoptotic or necrotic, anything like that, then you're only going to have one number per field of view. So you had 4 fields, so those 4 numbers will go here. And again, this is going to be the raw numbers. And then you are going to change this formula to reflect how you would get the percentage. This is not the correct formula. I want you to do it. I'm going to go through and help you with all the others, but this one, you have to figure out how to do it. Remember that to get percentage, you'll need the total number of cells, which are present back here somewhere, right here. So you need to be able to convert the formula to reflect that so you can get the correct percentage. Okay. How are you guys doing? Do you guys need a little bit of time to organize the data before we start talking about it and doing the analyses? Let me see. Can you raise your hand if you need more time? Okay, looks like a lot of you need a little more time. So I'm gonna stop the share for a few more minutes. And let's take about, let's say till 10:30, and you can go ahead and organize your data the way we just talked about, and then I will walk you through the analysis and how you're going to be doing it. In the module, there is a PowerPoint which goes through the basics of different types of statistical analyses and what's the pro and the con of each. Make sure you watch that. I'm not gonna focus on that in the interest of time since I do have to go to another meeting. But you can watch that for that basic info. I'm gonna show you how you're gonna analyze your data, and that's what I'm gonna focus on. Okay? So let's go ahead. I'm going to pause the recording again for a few minutes and you go ahead and organize your data. Okay? Start again. And share my screen.
I have a question because I forgot what you said, but for the starting confluency and the ending confluency for set 2B, set 2A, morphology, what are we supposed to put?
You're supposed to put the starting and confluence that you put down as question number 1 in morphology. And you may only have one number for each of your treatment, or you may have both for each of your cover slips. But that's what you want.
Oh, I see. That's,
You see it?
yeah.
Okay.
Thank you.
So that's your set 2. That's the one for the morphology analysis for the cover slip. And then set 1 is the 6-well plate with the 2 duplicates. Okay, so let's go ahead and get started. So now if we were to look at our end confluence by itself, let's say we had no information about this part, right? We didn't have a start confluence. And if we just compared these, what conclusions would you draw from it? What conclusions would you draw if you just looked at this one? The end confluence. So you're just focusing on this particular table.
Well, it looks like it decreased.
It looks like that there was a lot less cells in saffron and crocin. And what about if we just compared saffron and crocin with each other? Would you make any judgment on that? If one was more effective or not? Hmm?
Looks like they
Saffron's
have varying results.
What did you say?
Varying results because like Crocin has a higher cell count, for example, in like Viability 2 or set 1B, but then for the other ones it has lower cell count than
Saffron too, like one time it's at 40%, and then you have 25, 20, and look at the variation in the untreated even, right? It's all over the place. So your numbers may even be more variable, right? This is like a little bit more controlled data. So with this one, you can't really, you're not going to be able to see really good, consistent trends. So the only way that we can do that is if we normalize it to something. In our case, what we are normalizing to is the start confluence. And what it does is that it takes into account the variation that you had to begin with in your sample, because not every single sample started at the same confluence. Some started when they were already at 50, 60%, and others were only 30%. And that's an important piece of information that we're missing over here. So when we normalize a data, we normalize it to make it more standardized so that, oh, am I recording? Yes, I am. I think I am. Does it show you on your end if I'm recording? Yes, I am. So yeah, so what it's gonna do is that it's going to allow you to compare between the treatments and between the different sets, because it would have been all kind of standardized to this and kind of equilibrated to the same level. And so that's what we're doing when we do relative growth. We are taking that into account and we are normalizing it in this case to the start confluence. So what did we start with and where did we end? And so, of course, we've talked about this before because you have done it before in your own actual observations, right? In your own calculations, initial calculations. So a number above 1 means that over the course of the week, the cells increased in number or became larger, whatever, to cover larger percentage of the surface area. A number below 1 means that the cells either died or didn't grow as much, didn't replicate as much, so they were less confluent than you started with, or covered less percentage of the surface of your space than you started with. And so now when we do that, you see the numbers get a lot more closer together, right? So here I have that relative growth. And now I can begin to see a little bit of sometimes of a trend. Yeah. And I will say the Croson still shows a little bit of variability, but overall, now the numbers look a lot better. Yeah. Uh, Croson, if you remember, was kind of particulate, so it wasn't fully, uh, it wasn't like a solution. It was more like a suspension. So you can imagine that maybe when you're pipetting these different samples, one gets a little more than other. Having said that, you did make a stock of treatment, so it should not have been that variable. Okay, so now we want to start seeing the trends and visualizing them a little bit better. We only have 3 or 4 numbers, but imagine a case where maybe you have 100 numbers or 500 or even 50, right? It's going to be a lot harder to see the trends. So the way we do that is by taking averages and then looking at variability through standard deviations and standard errors. And that's what we'll do next. So we are going to take our average of the numbers that you have. If you only have 3 numbers, just choose 3 of them. If you have 4, choose 4. And then for standard deviation, we are gonna put STD and it will pop up 2 different functions. They have a standard deviation of population and standard deviation of sample. Which of the 2 do you think is the more appropriate version to do?
Sample.
Sample, exactly, because you're only looking at certain parts, right? You took a sampling of the population to collect for information on confluence. You didn't look at every single cell within the plate and calculate it for the entire population. So it's a sample, and then again, you take the same 3 numbers. So I just, you're selecting the numbers that you have in untreated for the first one. And then now, all of our numbers are for, in this case, have the same n number. But again, imagine a situation where your different conditions have different n numbers. Maybe for one particular treatment, you have n equals 10. Another one, you only have n equals 4. Another one, you have n equals 20. So when there are more individuals or more data points, then you can get higher variability, and that has to be taken into account. So we take into account and kind of again normalize the standard deviation or the variability in your sample by taking a standard error calculation. Standard error takes into account your n number. How many data points did you have to make up that average? In this case, our data points are 3, n equals 3. That's the number of replication I have on mine. Yours might have 4. So I'm going to do equals standard deviation divided by the square root of the n. The square root, so if you write SQR, the square root function will pop up. And then you write the number of replicates you have. Right now I have 3, so I will write 3 and press enter. So this is going to be my variability after taking into account the n number, the total number of replicates. Now I can go ahead and take these 3 formulas and drag them across to get it for the remaining samples. And so I'm going to end up with all of my numbers that I need. And then I'm going to do the things that I like to do to clean it up a little. So reduce them to 3. Okay. Now, the next thing we want to do is examine this. Now, looking at it clearly, visually, I can see there's a clear difference between my untreated and my treated samples. And within the treatments, they both seem to work pretty effectively, very similar to each other. That's great. But now I want to see if I can actually find out if quantify that significance and make sure that it is a real effect from the drugs that I'm working with. So what are some things that I can do? Which one are the ones that you are more familiar with in doing when we are working with such info? Hello. How do you typically look for significance?
If the p-value is less than 0.005.
Not 0.005, but 0.05. Yes. So one of the things that we've seen again and again is this p-value business, right? And so what we are looking for is a p-value less or equal to 0.05. What does that mean? When we say 0.05, what does that 0.05 represent?
I think it's like a difference of like 5%.
What about 5%?
That there's like a 5% difference from what you're trying to measure.
So again, I want you to think about it in like a layman term. So how would you explain it to somebody who doesn't, let's say, know any statistics or any such information? What does it signify?
That,
How does it know that
that's the amount
something?
of error they're willing to accept, like 5% of error.
Yes, so 5% error you're willing to accept, and what does that mean? What type of error?
It kind of shows like the correlation that you're it's hard to explain, I guess, actually.
Okay. So that's something that I want you to think about, right? And I'll tell you how I would explain it, and then you can tell me if that makes sense. Uh, so basically, when you're looking at 0.05, you're looking at that, yes, 5%. But what you're looking for is that there's only 5% chance that what you are seeing is just by chance or by error. And you have a 95% confidence that the change that you saw is actually from the treatment that you gave. Okay? So that's what it signifies. The 5% false discovery rate, that it was just an error or just by chance that we observed this change, and a 95% confidence that it is a real effect of your treatment. Does that make sense? And nobody speaks. So I have no idea if it made sense. Okay. So yes, so that's what we're looking for. And the typical way that we look at this is by doing a t-test. Now, a student t-test is a great way to look for changes and look for these significances. The problem with that is that it only compares two groups at a time. So there is no variation of it where you can examine 3 or more groups at the same time and look at all of them as a whole and decide whether there is something different amongst them. Because obviously, with different treatments, the more treatments you add, the more variability you will have, and that has to be taken into account too, as a whole, to see if there is some significance. So, to check for that, there are other tests available. The one that we are going to be using is Innova. And Innova comes in different flavors. It's kind of a grown-up t-test. Okay. So what it does is that it allows you to examine 3 or more groups at the same time. And not only that, it also allows you to look at multiple variables, independent variables at the same time. So you're not limited to just one single variable anymore. And you can do, 2 independent variables, 3 independent variables, multiple independent variables. It comes in various formulations where you can check for all those things at the same time and find out if there is something different between in that data set in one or more groups. What it will give you is that information. So it will tell you that in all these 5 groups or 100 groups or the various samples that you've given me, somebody is different than the others. somebody, one somebody or multiple somebodies, right? One or more samples are different, but it will not tell you where that is. It's not going to tell you that it's the saffron that's different or crocin that's different or both that are different, but it will tell you that something is different about one or more of these groups compared to others. And then you have to still go back and then run the t-test. And again, with t-test, you can only run two groups at a time. So you'll do pairwise, different t-tests. You'll do all the different permutations of it to figure out where exactly the change is, where exactly the difference is, which one is significant. Okay? So let's look at, we're going to do the ANOVA, but before that, let's look at the different t-tests that we can perform. What would be the different t-tests that you can perform to examine significance between two groups? What's the first one? So I'll get you started. So you can do untreated versus saffron. Yeah. What else?
Saffron versus crocin.
Yes, you can do saffron versus crocin, definitely. What else? Is there any other one?
Untreated versus
Untreated
course.
Huh? Say it louder. Untreated versus crocin as well, right? So you want to look at each drug independently compared to untreated and see if it is different. And then you want to look at saffron versus crocin together and look for that significance. Yeah? So there are two ways to do it. One is to use the analysis tool pack that is provided in Excel. If you don't have the full version of Excel and you only work on the online version, it won't be available to you, and you would need to go to the library for it. Now, for ANOVA, you do need to use the tool pack, and you will, if you don't have that available on your device, you will need to go to the library or on a friend's computer to do it. But for t-test, there is another way to run it, and that is a quick and easy way to create a formula for it. I hope you guys don't hear this like beeping car outside. I don't know what's going on. But here is how you would run that t-test, okay? And I actually prefer the quick and dirty version of the t-test compared to the other one. So for your t-test, you would write t-test and it will pop up. It will ask you, well, what do you want to compare? And so for first one here, I'm going to want to compare my saffron. So I will select not the label, only the numbers, right? And my three numbers that are there. Then it says array 2. That's what do you want to compare to? So I'm going to compare it to my untreated. The next question it asks is it a one-tailed or a two-tailed distribution? Does anyone know what that means? How do we decide if it's a one-tail versus two-tail distribution?
Is it dependent on the variables you're looking at?
Um, it can be. It's dependent actually on your hypothesis. It's dependent on what you started out with as your hypothesis, as your premise for your experiment. Anyone else want to take a stab? So if you think of a typical, data set, any data that you're looking at, it's gonna have like majority of the numbers that are falling within the main section of the graph, it will give you like a histogram, right? a bell curve so-called, and then you'll have some few numbers on either extreme, right? So in a one-tailed distribution, you have a clear idea based on your hypothesis as to how your drugs are going to behave. So you know, for example, or that that you are hoping that they behave. So in a one-tailed distribution, you are saying that my samples, when treated with the drug, are going to grow less, are going to die more, or whatever, right? On the other hand, in a two-tailed distribution, you know that they're going to be different, but you don't know how, or you haven't postulated on how. So you are saying that I think saffron treated samples will behave differently, but you are not sure whether they are going to grow more or die more, right? So the problem with that is it is fine. You can have it one-tailed or two-tailed, and depending on your hypothesis, you can choose which one is applicable. The problem with the two-tailed is that it's a little bit harder to find significance. You need to have more data points and cleaner data to see the significance, because that 0.05 gets kind of split between either this way or that way. But if you're doing a one-tailed and you're just focusing on one side, that they're going to grow more or they're going to die more, then all that 0.05 goes towards that one side. And so it's easier to find significance. In my case, I do think that based on what I've researched, saffron and crocin should both be effective as an anti-cancer effect. So I'm going to use one-tailed. But you will again think about your hypothesis and then decide which one would be applicable to you. And then the last thing that you want to select is whether it is a paired two sample equal variance or two sample unequal variance comparison. Now, a paired comparison is going to be when you have the same cells in culture and you take multiple measurements from it. For example, in this case, if we were comparing start versus end confluence, that's a paired sample. But we are not doing that. We have normalized it, and now we are comparing between treatments. We're comparing untreated to saffron and crocin. So it's no longer a paired sample because each one were in their own little dish and they were being treated independently of each other. And so they are going to be two samples, and now you have to find out whether they are equal or unequal variance. For that, you can look at your variability and you can decide. And based on that, the numbers are variable enough that I would say that it is two sample unequal variance. Again, this is the more stringent of the strategy. So unequal variance will require more numbers and more data points, cleaner data to show significance, but that's what you would do in this case. And when I look at it, I find 0.009, 0.00953. Is that a significant effect? Does this show that saffron behaved differently than untreated? Yes or no? Based on our premise that it's 0.05 or less p-value. So our p-value is, so this is our p-value. Our desired was 0.05 or less, and what we got was 0.000953. Is this significant? Yes? No? Maybe?
Yes.
Yeah. It's very significant, right? It shows that there is definitely a very clear correlation. And so I would go ahead and highlight that in some color so that I, as a reader of your assignment or another person working on your project, can easily see and spot that, yes, indeed, this was significant. Now, this is the why I like the quick and dirty number one. I can do this and at least the formula gets copied over, and then all I need to do is change it up change up the different sections to get what I need. And so in my second comparison, I'm going to be comparing Crocin to untreated and looking at that number. Is that significant? 0.001544. Yes, indeed. Good. Now I'm gonna do my third comparison. And so this one is going to be saffron versus Crocin. And I get 0.240699. Is that significant?
No.
No, it's above 0.05, so it is not significant. Okay,
I have a question.
yes.
So did you look at the standard deviation or the standard error to pick the equal or unequal variance?
I looked at the standard error, but you are actually going to be looking at variance and you're going to be looking at it from ANOVA. But this is the alternate way, in a quick and dirty way. This is the one that you would use. But we are about to do our ANOVA, and it will give you variance. So you'll be able to run that as well.
Okay, thank you.
Okay, so we got our t-test and we see where the significance lies, and we know that it really does exist, right? And that's cool. Now we need to confirm that, ANOVA shows that actually when I look at all the groups together, it does show significance, that there is something different there. So if it shows that if there can be times where your p-values individually for t-test shows some difference, but when you run the ANOVA, overall there is so much variability that it says no, it doesn't mean anything. And in that case, that's what it is, that if you look at the entire group as a whole, it's not going to be significant. It's rare, but it does happen. Now, to run the ANOVA, we need to have the tool pack. If you don't have it, and you can check to see if you already have it downloaded, you would look under data and you will see this data analysis tab. If you do not have the tab, you need to go to File. If you have a non-Apple computer, you go to File, and then you go to Options. You open that up, and in here, you're going to see add-ins. So you click on add-in, and up down here, it will say Manage Excel Add-ins. You click Go, and you make sure that you have the analysis tool pack, and click OK. If you have an Apple computer, I think you go from the tools on the top toolbar. Like there's, I think, tools or something, and then you get to options and do the rest of it the same way. Okay, so to run the ANOVA, you're going to click on the data analysis tab, and you will go up. Now, there are two different ways that you can run it in Excel. You can run the single factor or two factor with or without replication. A single factor means that there was one independent variable. A two factor means there were two independent variables. For example, if I treat with saffron and I treat with low versus high at different concentrations, even though I have multiple groups and they are all being treated with, they're all still being treated with saffron, even though it's different concentrations and different groups. So you would still have one independent variable. But if you have an independent variable of treatment, like I treat saffron, but with low, medium, high, so I'm still treating with multiple concentrations. But then I also collect samples at different time points. So I collect them at 12 hours, 24 hours, 36 hours, whatever. Then I have two independent variables. Treatment, whether or not the cells got saffron, and the time point at which the sample was collected. Do you get that? So that would require a different type of analysis, which is called two-factor analysis. Now with two-factor, you cannot have different n numbers. So if you don't, the better way to do it is to do it with replication and use all your individual data points. But if you have 100 data points in one and only 20 in the other, then what you do is you take the averages of each and run it without replication, okay? But in our case, what do we have? Do we have a single independent variable or two independent variables? You guys there? Did I put everyone to sleep? Why? Why do we only have one?
Aren't we? Oops. Are we comparing untreated versus saffron? And so like we're only have like one independent variable, the change of whatever the treatment will be.
But what about frozen? That's a different treatment, no? Yes, no, maybe. So technically, they're two different drugs, so they're two different variables. However, in our case, we're going to still make a case that it's a single factor because saffron contains frozen 2, that's the active compound. So essentially, we are treating with a mixed crocin, you know, not a pure crocin versus pure crocin. That's our background on it, okay? So we're gonna still run single factor, mostly because it's easier for you guys to run that too. So we're gonna click on single factor and it will say, okay, what do you want to compare? Input range is where you need to put in what you're comparing. So I'm gonna click on this arrow. I'm gonna come up and I'm going to select the headings. So where it says untreated saffron crocin and all my individual numbers for relative growth. So you use the normalized data, okay? And then I will come back, it will have those. And then I will click on this little box that says labels in first row. So it knows which group is which. I will keep the alpha at 0.05, but you don't need to worry about changing it, but you do need to know that that is not entirely accurate when we are working with more than 2 groups. When you have multiple groups, again, you need to take into account the variability. And one of the things you need to do is to correct this in your p-value that you are going to consider significant. So it becomes harder to find significance again. And so with the 3 groups, it's essentially n-1 that you are going to, you know how when you did chi-square, you did the n-1, same idea here. So in n-1, that would be 2 groups. You would divide the 0.05 by 2 to get the actual acceptable p-value. That is called the Bonferroni correction. You need to just know that it exists. You're not going to worry about it. We're just going to use the 0.05 as our baseline. And then we are going to put in, by clicking on the output range, where we want the data. You want all the data on the same sheet. So I'm going to go ahead and select where I want it. You just select one well to indicate this is where you want the data analysis to start. And click OK. And now you get 2 charts. You want to confirm that your counts match your replicates. So I had 3 values. There is says 3. That's good. You should double check that your average matches the average given by Innova. These are internal checks to make sure you added the stuff correctly and that your groups are labeled correctly. And right here, for those of you who asked about the variance, you can see the variance right here. And you can see that it was indeed unequal. At the bottom, you will get your ANOVA answer. And what you're looking at is this, you can look at the F critical versus the F value, obviously. Again, this is your degree of freedom, which is 2. And now you look at the p-value, it's 0.0000300289. Is that significant? Is that a value that shows that somewhere in those 3 groups that something is different?
Yes.
Yes, indeed. So you will highlight it. Only highlight values that are significant, so they are easy to spot and we know what is being shown. Okay? We're almost to the end of this first analysis, and then the rest of them you're going to be doing on your own, the same idea. I will tell you a little bit of something that you need to think about when you do tribe and blue though. Okay, so now what is left in here is the graph and the conclusion. Okay, so let's make a graph. What type of graph would be appropriate for this analysis? To show to the reader what we saw, the trend that we are seeing. What would be a good type of graph to make? Scatter plot with all the numbers? So you want to do like that? Like that? So again, you know, when you have small amount of data, sure, that could work out. It didn't give the labels correctly though, so we would need those. You could do that. What would be another way or that you can show it? And this is cool because it kind of shows the variability between the data too. So yes, definitely you could create a scatter plot. What other things could you do?
Bar graph.
Bar graph, and then how would you show the variability in that case?
You compare the 3 different variables next to each other with the different values.
No, but how do you show the variability within a treatment, like within untreated or saffron? Yeah, we can use the error bars. What should we show in the error bar? Standard error or standard deviation? What would be the better number to show? Standard deviation, remember, does not take into account the n number. So it's not a normalized standard error.
So the standard error.
Exactly. Standard error would be the better way to show it. So to make your graph, what you're going to do is select your labels against the averages. That's one way. Again, you can do a scatter plot the same way, and it will show the variability too. You got your basic scatter plot. I'm going to keep it up here so I can show you the things that need to be done with it, right? Now you need to format it according to what we need. You're going to add access titles. You can add the chart title or you can remove it. Because we are preparing things for poster, it may be a good idea to show. It's up to you. That's your choice. Then you need to remove the grid lines. You need to add the legend. You need to add the error bars. When you get to the error bars, you're going to click and go to more options. You're going to have both sides. You're going to do custom, specify value. Again, just like the Innova tool pack, this is another thing that you cannot have access to. In the online version of Excel, you'll need the full version. Everything else for the graph, you should be able to do without it. Okay, so now I got my error bars on there. Now I want to start formatting it. So I'm going to click on the x-axis. I'm going to put a solid line in black that's visible. I'm going to remove this title because we already have the treatments listed. Same thing here. I'm going to put a solid line. Over here, I also want to make sure that the numbers are not so busy. So maybe I just keep 1 decimal point now, right? And if you wanted to, you could put tick marks or labels or something else. This one is going to be my average relative growth, right, between the samples. You then want to make sure there is no border, no line for the border in outer or inner square. Okay, so far so good. Now we need to modify this. Again, this is for a visual product, so you want to make sure to format it accordingly, right? So for example, for your titles and everything, you want to make sure it is the font size is good. So you want the font size of at least 12 or 14 and bold when needed. So this is 10, so I'm going to make it 12 or 14. 14 is good. I'm going to make this one 12. I'm going to give it a little more breathing room. I'm going to make my titles 14. And I'm going to make them bold so they're nice and visible. I might even want to put the actual concentration in there, right? And next, I'm going to go to my bars. I'm going to put a border around them. So outline, put a black outline. Then you double click until you just have that first bar selected. And you change the fill color. For your untreated, you want to kind of keep unobtrusive color, right? So something that doesn't really bring it out too much. That's not our focus. Our focus are these other treatments. So I clicked on the saffron now, and I'm gonna choose the color for that. And then I'm gonna choose the color for crimson. And if you look over here, here is my legend. I'm gonna make sure it's also in 12. Make it so that it fits right where, oops, not there. And you can leave it like that in the corner here. It fits, it looks good. You can put it like that, right? You can show it whatever way you find useful. Okay. And lastly, you want to put in your some little, and that is better done in PowerPoint anyway, but you want to put in a little text box with an asterisk to indicate that there was significance compared to untreated control. Okay. The last thing you're going to do is make a text box, and then you're going to copy and paste the text box and this figure into a PowerPoint as well. But to insert a text box, you can just go in here, put a text box in, and you're going to write the end conclusion from this graph or this analysis from relative growth. And your conclusion should be biologically relevant. So you're not going to write that saffron and crocin show significant difference, P is equal to 0.000289 or whatever, right, compared to untreated control. That means nothing to a layperson. What would be a good conclusion to draw from this?
The growth average between untreated and saffron, their basically untreated average had more growth compared to the saffron treatment.
What's our experimental treatment? Remember, experimental treatment of saffron and crocin, that's our comparison. That's another thing. So you're on the right track, but you don't want to talk about untreated. You want to talk about your experimental treatment, right? So you can talk about how both saffron and crocin show similar inhibition, right? Or similar anti-cancer activity through reduced growth in response to a 1-week treatment or whatever, right? Or saffron and crocin both inhibited growth of the cells at the same rate compared to untreated in vitro. So that takes into account the fact that saffron and crocin both showed significant difference compared to untreated. So they did work and did have anti-cancer activity. And how did they work? They reduced the growth of cells. Right now, we don't know if they reduce the replication rate or if they kill them. We just know that there are less cells. So they reduce the growth of the cells. And then the fact that we said saffron and crocin both worked similarly takes into account that our saffron versus crocin value was not significant when you look at the p-value. Does that make sense? Something along those lines, right? And that will then take into account all the t-tests and the whole analyses and talks about both the saffron and crocin. So last thing is, we are going to make sure that everyone needs to be doing some part of the analysis from beginning to end. We all did relative growth together, so you can make this text box with this conclusion, let's say gray. Okay. Now you have 1, 2, 3, and in this 2 there are a couple of different ones. Then 3 and 4, right? So every student, even if you're not comfortable with Excel, should do something. Out of the remaining 6, at least 2 should be done by 1, if not 3, and divide equally. If one person does the analysis, I want you guys to pick 2 primary colors, let's say yellow and red. And then if one person does it, the analysis from beginning to end, so all the averages, standard deviation, normalization, the statistics, and the graph and conclusion, all are done by one person, you can make that yellow. If the second person does it, you make this text box red. And if you guys work together on a particular sheet or analysis, let's say you work together on nuclei per cell count or on dying cell percentages, then you make it what color in that case? If I use yellow and red?
Orange.
Orange, exactly. So Ms. Lindemann can easily see when one person has worked on, what one person worked on, what the second person worked on what you did together. And everyone should do at least one set of information from beginning to end all by themselves. And then you can work on the rest as a group. Okay, so we should see at least one text box that's yellow, or one primary color, one text box that is the other primary color, and the others can be then orange or whatever the combination color would be. Okay? You guys good so far? So just by going through this either right now with me or in the recording, you should be able to get this relative growth done without a problem. Everyone should have that the way I've done it, showing this information. Now, let me just tell you one little thing about the rest of it. So for this one, this is our normalized data, which is the percent cells, because obviously, if you look over here to the raw data, the numbers are all over the place, depending on the treatment. Sometimes you had a lot of cells, sometimes you had very few cells. But when you convert it to percentages, they all get normalized. And now you can compare between the different groups. Now, you are going to look at it and you're going to decide what makes sense. So in my case, because I'm looking at this anti-cancer activity and looking at whether these are able to kill my cells, I'm going to be focusing on percent dead. And if I do that, I'm going to then change the heading to just reflect it and say percent debt. Next, I'm going to organize the data according to what I'm using. So I'm going to take the numbers up here and copy them down here by writing equal sign. And because I'm doing debt, it will be the blue percentage. If you're doing living cells, so if you're looking at cells that were left behind that were resistant to your drug, then you would be looking at percent alive, and you look at clear cells. So I'm just going to say that I'm looking for my cells that were dead, and then here as well. And then I'm going to drag this across to get my remaining numbers. And once I have them, I'm going to take my average, standard deviation, error. When I do the error, what will be my square root of this time? What's my n number this time?
4.
4. Perfect. So you can even put that right now while we are together, equals whatever standard deviation divided by square root of 4. Okay? And that's what will be over here. And then you'll run the same ANOVA, t-test, and whatnot. Over here, you're going to decide, again, based on your hypothesis, whether you care about the cells that are dividing, or do you care about the cells that are dying? And depending on that, you will only use the information for that site to run the ANOVA, t-test, and the graph, and make the conclusion. Everyone's going to do the first two, though. Everyone's going to do the relative cell count and the nuclei per cell. Again, you're going to do the standard, the averages, standard deviation, and standard error. In this case as well, you are going to have n equals what? For these ones, I have 4. If you only had 3 pictures to work with, then your n is going to equal 3. I'm going to copy it over here, and here, and here. For cell and nuclear measurement, you will put in the numbers here. You will then use your one of the two between dividing and dying. That's why it only gives you one place. So you decide between dividing and dying which one you want to show. And same here, you decide between cell body diameters versus nuclear diameters, which one is showing something interesting, shows a difference. And either way, you need to show at least one, but you can pick which one of the two you want to focus on. And in this case, remember, after average, you're going to, the first thing you're going to do is convert it into micrometers. So you are going to take the formula according to your calibration. So if your calibration was 9.9, then that's what you use. If your calibration was 11, then that's what you're going to use. And you will put your numbers in pixels, and then it's going to calculate the numbers in micrometer for you. And then you're going to do the standard error. In this case, what is it going to be divided by? Standard deviation divided by? Tell me, square root of?
10.
10, exactly. Okay. So hopefully by the time you're done with all of them, you would be friends with Excel. Okay. So you do that, and then you do the same thing for your interesting observation. All the data, once you have all your graphs, you're going to copy just the graph and the conclusion to a PowerPoint. So you will have 7 total slides in the PowerPoint that you will turn in along with your Excel sheet. These are the 7 that you're doing. Okay, it shows you right here. Each one is worth 10 points, essentially. Questions? I have to go in 3 minutes, so but I can take a few questions.
I was curious, so you said we're supposed to like make a PowerPoint as well with Excel. So what stuff are supposed to be in that PowerPoint? Like everything?
No,
Uh,
so let me show you. So this is my complete analysis from your first one, right? This is what we did. Let me make it a little smaller. So when I make a PowerPoint, let me open a random PowerPoint, right? For each one of the things, once you're done with them, you're only putting in, let's take it over here. Oh, somebody has sent me this. Let's say this is my PowerPoint, right? The only thing that I'm gonna put in here are this, and I'm gonna copy this over.
your
screen.
screen's not sharing.
Oh, sorry. Thank you. Where did my share go? Boom. 1 second, share. Okay, so you see it now?
Yes.
Okay, so you're gonna copy this, Control C. You're gonna put it in here, but remember, keep those in the Excel as well in case you need to change something. And you're gonna paste it as a image, right? And then you can make it big, small, whatever you want, right? And what you're gonna go along with this is gonna be your conclusion. Right here, this one. And the color that you want it to be in. And you're gonna put that in, you can put that in as a title or as a endpoint at the bottom, but that's what's gonna go up here. In my case, I just wrote it like a title anyway. So I'm just gonna, and it's gonna be in the right color, so I'm able to see who did what. See it?
Okay, so basically we're putting all of our actual data that you see in a graph or something and the caption in the PowerPoint.
Eventually, yes. So
And
all
each
your data analysis is in Excel and the graphs and everything, but then the PowerPoint only contains the graph and your conclusion.
Okay, and each conclusion in each graph will have a separate slide.
Yes.
Okay, that makes more sense. Thank you.
So you will have a total of 7 slides. Okay? And you see how I put the asterisk there? This would be for me to show significance. Between untreated and these treatments. Cool? So yeah, so that's what you'll be turning in. Is that good, guys? Okay, I got you go. And make sure you please turn in your sheets. I'm going to be in my office for office hours, and then whenever I get a chance, I'll treat them. So I need that information submitted so I can treat them appropriately and label your names, your group IDs correctly. Okay?
Hold on. So I was a little confused in the calculations because I thought I did it right, but then you were